Welcome to STUDYHIGH.Pk and subscribe, keep in contact for more videos and lectures. Now I am going to start our lecture 4 of chapter Lipids, Phospholipids and its types part 1. In this lecture, I will discuss the structure, the function of different types of phospholipids present in our body, their location, how they involve in definite reactions. We will also discuss the different type of diseases that are related to these phospholipids. In previous lecture, I have explained in detail the different types of eicosanoids, their role, their functions, prostanoids, how they can cause inflammation and asthma, and how the different type of drugs like aspirin, ibuprofen, and other type of drugs can be used in treating the patient suffering from inflammation and asthma. In this lecture, first of all, I start discussion from triglyceride, their importance in our body. I do not waste my time in explaining how triglycerides are formed, as I have already explained the structure how lipids are formed in my first lecture. Triglyceride consists of one glycerol molecule. Glycerol consists of three carbon. That it glycerol is a trihydric alcohol, and its three alcohol hydroxyl group react with three fatty acids to give rise to triglyceride, or another name triacylglycerol, because this consists of three fatty acid groups that can react with hydroxyl group of glycerol and results in formation of ester linkage between them. Some tissues may also consist of two fatty acids and are called diacylglycerol. Those tissues are body cells which consist of two fatty acids and one glycerol are called diacyl, monoacyl glycerol are also present. But I am going to discuss the importance of triglycerides for a human body. If this is a human like you and like me, it engulfs a lot of lipid. Or what happens to extra lipid that this person eat or drink? You must think excess lipids should be stored. And where lipids are stored in a human body? Lipids are stored in adipose tissue. Just assume this is a adipose tissue. And in what form lipids are stored in our adipose tissue? Lipids are stored in adipose tissue in the form of triacylglycerol. So triacylglycerol is the form in which lipids are stored in a human body, and when we need them back, this will be broken down and released from the adipose tissue and enter in our bloodstream. Then they may go to the mitochondria for the production of energy by passing through different type of cycles like Krebs cycle, electron transport chain. So if someone asks you what is the storage form of lipid and how triacylglycerols are stored or helpful for our body, triacylglycerol is a form Triacylglycerol is a storage form of fatty acid. Second important thing that you must know about the structure of fatty acid is that it consists of three carbons 1, 2, 3. They are different from each other. If you remember, in glycerol, every fatty acid that attaches with this glycerol third carbon, the fatty acid was sometimes what will happen in formation of phosphatidic acid. This fatty acid has been replaced by phosphate group. Phosphate group is always attached on carbon 3 of glycerol, where in case of sphingosine, it will always attach on carbon number 1. So glycerol structure shows that its phosphate cannot attach on carbon 1. Carbon 1 is stereoisomerically different from carbon number 3. If a stearic acid attach at carbon number 1, then in formation of a definite type of triacylglycerol, stearic acid cannot attach on carbon 2, or specific acids will be attached at specific carbon for formation of a specific product. As I have explained you in structure of phosphatidyl in carbon number 1, stearic acid attaches, whereas on carbon number 2, arachidonic acid attached. So structure and carbons of triacylglycerol all carbons are different from each other. Now we will discuss the phospholipids and its different types. The first, the most basic, all these phospholipids will be derived from the triglycerols. A triglyceride is a precursor of all types of phospholipid. First of all, I want to just to show you the structure of phosphatidic acid, which is the simplest phospholipid. In phosphatidic acid, carbon number 1 is attached with one acetic acid through ester bond of glycerol. Carbon number 2 attached with second acyl fatty acid, whereas the carbon number 3 of the glycerol connects with phosphate group. This is a phosphate group consists of 4 oxygen atoms. This is the simplest structure or simplest phospholipid that consists of one phosphate group at carbon 3 of glycerol. This is the further derivative of different type of phospholipids. Some are involved in performing functions or roles in as second messengers. Some involved in performing different type of conduction system in nerve fibers and central nervous system. The first, the most abundant type of phospholipid that we will discuss first is lecithin. What is structure of lecithin? In lecithin, all the structure remains same. Lecithin also consists of two fatty acids connected at carbon 1 and carbon 2 of glycerol. But the only difference is that this phosphate is converted into phosphatidylcholine, or this phosphate is modified. The modification of this phosphate group results in formation of different types of phospholipid. This is the structure of choline that consists of three methyl groups connected with carbon to N positive nitrogen atom. The hydroxyl group connected at this carbon reacts with the hydroxyl group of phosphatidic acid and results in formation of another ester linkage between the choline and phosphatidic acid, results in formation of phosphatidylcholine. And second name of lecithin is that phosphatidylcholine. So there is another bond formation, ester bond formation between the P double bond O phosphate group and choline. As a result, we will obtain a phosphatidylcholine, the most abundant phospholipid of cell membrane of our body. Different cell membranes of body, most abundant component is that our cell membrane contain is phosphatidylcholine or lecithin. Two important functions that are performed by lecithin or phosphatidylcholine in our body. First, phosphatidylcholine is a source of choline, and choline, like acetylcholine, is act as a neurotransmitter outside the central nervous system. We have also studied about acetylcholine, which act as a neurotransmitter in conduction of impulses from between the synapses, between the axon ending of one neuron to the dendrite of other neuron. These act as neurotransmitter released by this synaptic node, and they cause or trigger action potential in the next neuron. So the lecithin consists of phosphatidylcholine, and it can be act as a source of, or it has a large amount of choline that can act as a neurotransmitter in conduction of nerve impulses. This is the first role that is performed by lecithin in our human body. It can store and then also provide on breakdown the different type of choline molecules that will act as a neurotransmitter in conduction of impulses from one neuron to another neuron. The second, the most important function that is performed by lecithin is in our lungs. They involve in protecting our lung from the disease respiratory distress syndrome, in which there is the lungs collapse with each other. So what is the role of lecithin in preventing the lungs from collapsing? If the carbon 1 and carbon 2 consist of palmitic acid that consists of 16 carbon atoms and is unsaturated fatty acid, as it also consists of one double bond. Palmitic acid so is a monoenoic as it consists of one double bond. So if carbon 1 and carbon 2 of phosphatidylcholine consist of two acids that are palmitic acid, then we will call it dipalmitoyl lecithin. This dipalmitoyl lecithin is produced by the cells of our lung called pneumocytes. They produce dipalmitoyl lecithin. 90% of surfactant that is produced by the liver cells consist of dipalmitoyl lecithin or other lipids, whereas 10% consist of proteins that reduce the surface tension and help the lungs to inflate easily without much resistance. So help the lung in reducing tension so that the lungs can easily inflate. And second thing, they reduce the surface tension so that the lungs do not collapse with each other. If the lungs collapse, death of the newborn or the patient can occur. This is the structure of lecithin, and lecithin consists of choline, and choline can act as a neurotransmitter in conduction of impulses. Second important function performed by lecithin is that they can also act as a surface reducing agent or surfactants in our lungs and prevent the lungs from collapsing. Now I am going to discuss two other important phospholipids, cephalin and phosphatidylserine. The structure of cephalin and phosphatidylserine is very easy. You must need to remember the structure of serine and ethanol amine. Cephalin consists of ethanol amine that is connected with this phosphate group, whereas phosphatidylserine consists of a serine amino acid. This is the structure of serine. It is an amino acid that consists of a carboxyl group connected with this carbon, and amino group is also connected with this carbon and other CH2 and hydroxyl group. This is a serine amino acid. Through its hydroxyl group, it is connected or form a ester bond like choline, as choline did. As a result, the resulting product is phosphatidylserine. When ethanol amine, this is the structure of ethanol as it consists of two carbons, and NH3 positive is connected at one side of carbon and second carbon is connected with the hydroxyl group. This can also form a bond with this phosphate group. The modification of this phosphate group results in formation of phosphatidylcholine. Then further modification can result in formation of cephalin. The second name of cephalin is phosphatidylethanolamine. Phosphatidylserine has a role in causing apoptosis, programmed cell death. When a cell causes or induces changes in its own metabolism or different type of reaction, it induces its own death. When a cell suicide, the suicidal death of some cells of the body is called apoptosis, and phosphatidylserine help in causing apoptosis of the different type of cells of the body during different type of conditions. If a body facing starvation or different type of condition, if some cells are abnormal and do not be needed by the body, apoptosis, programmed cell death occurs. And phosphatidylserine also helpful in causing apoptosis. Phosphatidylethanolamine is also a component of cell membranes of cells. Now I am going to discuss the little conceptual and the most important sphingomyelin that has an important function in cell signaling and cell recognition. In our first lecture, I have explained you the basic components of sphingomyelin that consists of fatty acid. The alcohol that involved in formation of sphingomyelin is sphingosine, not glycerol. And the third component is phosphatidylcholine. Phosphatidylcholine or phosphate group derivative. The phosphate group or phosphatidylcholine is involved in formation of sphingomyelin. First of all, I will show you the structure of sphingosine. What is the structure of sphingosine? You know the structure of glycerol, but sphingosine is first lecture I informed you about the structure or components of sphingolipid that consist of fatty acid. The second component that is common to all, alcohol, and third component is phosphate group. that may be modified with different type of bases like choline, serine, ethanol, amine. The alcohol that is present in other types of phospholipid is glycerol, whereas the alcohol that is present in sphingolipids or sphingomyelin, that is a subtype of sphingolipid, is sphingosine, not glycerol. Whose sphingosine and sphingomyelin structure is different from glycerol containing phospholipid. The sphingosine also consists of three carbons like glycerol. At carbon number 1, it also consists of hydroxyl, but in carbon 2, rather than containing hydroxyl and connected with acid, it consists of an amino group and it forms or reacts with acidic group at carbon number 2 through the amino group, not through hydroxyl as occurs in other type of phosphoglycerolipids. So in this case, this sphingomyelin is different from other types, whereas carbon number 3 of sphingosine is also different from glycerol containing phospholipid. Its hydroxyl group remain intact, and on other side of carbon there is a tail, the alkyl tail, that is unsaturated and consists of one double bond, and this R group consists of approximately 13 carbon atoms. So total 15 carbons are connected with carbon 3 of sphingosine, whereas hydroxyl group remain intact, and at carbon number 2 there is an fatty group or C double bond O that is connected with this amino group with sphingosine through this amino group, not hydroxyl. This is an acetic acyl group fatty acid that connected with carbon 2 of sphingosine through NH. So this is amide linkage between the fatty acid and NH of this sphingosine molecule. Whereas in case of sphingomyelin, the carbon 1 of hydroxyl phosphate group attaches at carbon 1 of sphingosine, whereas in glycerol, the location of attachment of phosphate group is carbon 3. This is a third difference between the sphingosine or glycerophospholipids and sphingophospholipid, that the phosphate group in sphingolipid attaches at the carbon 1, whereas in glycerolipids or glycerophospholipids, the phosphate group attaches at carbon number 3. In case of sphingomyelin, carbon 1 of sphingosine is phosphated or phosphorylated, and this phosphate is further attached through another molecule of choline. So in sphingomyelin, at carbon 1 of sphingosine, there is a phosphoryl choline. There is attachment of phosphate group that is further attached with a choline molecule, as you see in phosphatidylcholine, lecithin. At carbon 2, a fatty acid group attaches through amide linkage, C double bond O NH. This linkage is called amide linkage. At carbon 3, there is an alkyl 15 carbon containing unsaturated alkyl group that attaches at carbon number 3, whereas the hydroxyl in case of sphingomyelin or sphingolipids remain intact. When sphingosine molecule react with fatty acid only, then the resulting compound is called ceramide. Ceramide is the compound that found between the sphingosine and fatty acid. So the addition of fatty acid and sphingosine or reaction of sphingosine and fatty acid results in formation of a molecule that is called ceramide. The structure of sphingomyelin is little difficult. No, I think you can easily draw. First you have to draw 3 carbon atoms like glycerol. At carbon 1, the phosphate or phosphoryl choline you should have to attach on carbon 1 rather than carbon 3. At carbon 2, you have to attach an fatty acid group through amide linkage. At carbon 3, hydroxyl group remain intact, and other is a sphingosine tail that consists of R, C double bond, total 15 carbon atoms. This is sphingosine tail. This is the structure of sphingomyelin. The most important function of sphingomyelin is in nerve fibers. In nerve fibers, if you remember, nerve fibers consist of myelin sheath. What is function of myelin sheath? This myelin sheath provided insulation as it cannot conduct impulses. First thing, myelin sheath provides insulation to nerve fibers and protect our neurons and nerve fibers. So sphingomyelin provides protection as myelin sheath. A second thing, it also increases the speed of conduction of different type of impulses as they can jump from this insulated area, and jumping results in jumping nerve, jumping action potential. As a result, speed of nerve also increases because of presence of sphingomyelin. Now we will discuss the degradation of sphingomyelin. How sphingomyelin is broken down in our body. There are two important sites of breakdown of sphingomyelin. One is liver and second is spleen that involve in breakdown of sphingomyelin, and two type of enzymes involved in breakdown of sphingomyelin. One is sphingomyelinase. Sphingomyelinase cause the breakdown of bond between the carbon 1 and phosphodiol and the phosphodiecholine. It will result in breakdown or removal of this component. Phosphodiecholine will be removed by the action of sphingomyelinase enzyme. And second type that involves in breakdown of sphingomyelin, as sphingomyelin consists of three components. One is phosphodiecholine and second is fatty acid that connects at carbon 2 through amide linkage. So second enzyme that involved in breakdown of this sphingomyelin is ceramidase enzyme. As you know that the reaction of sphingosine with fatty acid involved in formation of ceramide, and ceramidase enzyme cause the breakdown of this bond. Two type of enzymes about which you must know: sphingomyelinase and ceramidase. Sphingomyelinase break the bond between the phosphodiecholine and carbon 1 oxygen, whereas ceramidase break the link between the amide linkage, between fatty acid and amino group of sphingosine, and results in breakdown of the sphingomyelin into its original component. And if there is a deficiency of these enzymes, the sphingomyelin will not be broken down into its component. As a result, they will accumulate in liver and spleen. Accumulation of sphingosine into liver is called fatty liver. Accumulation of lipid in liver results in fatty liver disease, and second in spleen. They will also disturb our mental retardation, mental functioning, nerve system, and different type of diseases. But they can also cause death in children as well. This disease in which there is deficiency of sphingomyelinase or ceramidase is called NMDCAT and PK disease. Because of deficiency of these enzymes, there are two types of NMDCAT and PK disease, A type, B type. So sphingomyelin is rather a difficult topic. I try to explain in a better way. I explain you what is the structure of sphingomyelin and how it is different from glycerophospholipid. How sphingophospholipid differ from each other. In glycerophospholipid, phosphodiecholine connect at carbon 3, whereas sphingophospholipid it connects at carbon 1. In glycerophospholipid, at carbon 2, fatty acid attach with through O glycosidic bond. Through O bond, whereas in sphingosine or sphingomyelin, the fatty acid connected at carbon 2 of sphingosine through amide linkage, whereas the third component or third carbon of sphingosine is connected with an alkyl group that is unsaturated, consists of 15 carbons, and hydroxyl remain intact at carbon number 3. So this is the structure of sphingomyelin and its function in cell signaling, cell recognition, what are the enzymes that involve in breakdown, and what disease is related with the sphingophospholipid or sphingomyelin, and different type of effects: liver enlargement, spleen enlargement, nervous problems, and mental retardation in patient. So hope you best of luck. This is our lecture 4. In next lecture we will discuss the remaining phospholipid. Allah Hafiz. Like, subscribe, share. Thank you.